Ecologists study the makeup of biological communities and ecosystems. They look at how living and non-living factors affect the abundance and distribution of organisms. They also investigate the effect of changes in the environment on the organisms in a particular ecosystem. To do this, they must be able to measure how many organisms there are and how those organisms are distributed in the first place. The simplest way to count the number of organisms is to use a sample area called a quadrat. We often use a square frame laid on the ground to outline our sample area. People refer to these frames as quadrats too. A quadrat with sides 0.5 m long gives you a 0.25 square meters sample area. Quadrats are used to investigate the size of a population of plants. They can also be used for animals that move very slowly, such as snails or sea anemones. You use the same size quadrat every time, and sample as many areas as you can. This makes your results as valid as possible. Sample size is very important. You must choose your sample areas at random. This ensures that your results reflect the true distribution of the organisms, and that any conclusions you make will be valid. There are a number of ways to make sure that the samples you take are random. For example, the person with the quadrat closes their eyes, spins around, opens their eyes, and walks 10 paces before dropping the quadrat. A random number generator is a more scientific way of deciding where to drop your quadrat. You need to take several random readings, and then find the mean number of organisms per m squared. This technique is known as quantitative sampling. You can use quantitative sampling to compare the distribution of the same organism in different habitats. You can also use it to compare the variety of organisms in several different habitats. Sampling is also used to measure changes in the distribution of organisms over time. You do this by repeating your measurements at regular time intervals and calculating the mean. Finding the range of distribution and the median and mode of your data can also give you useful information.